We're now going to move on to our next presentation. We have the pleasure of having here with us Dr. Roser and the very finest team that we have here on the peninsula and even beyond, working here with us. Do we have her presentation? Okay, yes, here it is.
¿Tienes puntero? Bueno, está aquí.
Sí, déjamelo. Además es el mismo modelo.
Sí, es el mismo modelo. Vale.
¿No te has atrevido con mi apellido?
Es que no me acuerdo de él. Lo voy a decir si es. Urreizti. Más o menos. Bien, el grupo de CIBERER de Sant Joan de Déu, en Barcelona.
So they're doing an amazing work in Barcelona, not only for rare diseases, but for many of those. I usually try to lean back from the microphone, but, well. First thing I wanted to do is to introduce myself. You everybody know Kristen, but you don't know me or us. Some of you, you know my face, but others, this is just new. I wanted to introduce who we are, what are we doing, and why are we here. So you can see me here in San Juan de Dios group. We put ourselves a label, which is the Bandage group. It was just to give a name to this collaboration, this partnership, which is effective. It's not that I'm just working with you tomorrow and the day after tomorrow with another one. No, we are very working together. We are very well structured and labels work. So this is the University of Barcelona. The group before led by Susana Balcells and Daniel Greenberg, and now it's on charge Dr. Ravionet. So basically Raquel is going to be the leader because the other two doctors are going to retire pretty soon. We also have Roser Corominas, expert in cell models, and also the youngsters, which are the ones that basically are doing the work, which is Mónica Centena, Ina Prats, and Juan Diego Gutiérrez. I started working in this group years ago, in the year 2019. The ones that you know me from before, you said you were in the University of Barcelona because you met me there, and my heart is basically there, and most of my work is still there. But I moved to San Juan de Dios Hospital in Barcelona, and has been a very interesting change for me because this allowed me to be more in contact with a biochemical team directed by Dr. Artuk and amazing clinicians and amazing people. It's Dr. Serrano. And I didn't have space for more photos, but there's an amazing team of neuropediatrics that they are focused on the patients, and they've taught me many things. And we love team building and team playing, so we worked with Isaac Canal's doctors. He's just a son of science of the University of Barcelona, and we are learning so much from him, from cell models. And as all of these experiments at a informatic level and mathematical level is just another arena, it's very important to have people that have the knowledge. So our support on this more logistic side, we have Pedro Seoane in the team in the University of Malaga, and we're also part of the Identity, which is the working group of organoid and cell models, which is an effective working group that was born from the CIBERER. And also Pablo de la Vida University is also participating there. All of this comes from this umbrella, which is the CIBERER. Paco will tell you a bit more about this, but it's basically how to study how rare diseases work in Spain. We have a virtual entity because the CIBERER, it's the investigation of rare diseases in network, and this gives us a solid structure of communicating, working together, and also to obtain financing, but also to have a network structure. So under this structure or this umbrella, we have this group. And how did we start? We started with a coffee, as every good story. We started in a genetics course in which an an informatic guy, is enrolled. He had a daughter that she basically had this label of the obese C syndrome, and Carlos came and said nobody is studying this in the planet, and this gives you this feeling of isolation, of being lonely. So in that moment, we started having these new techniques of new sequencing. So with the support of Carlos, of Dr. Obi, we decided to start studying this, thinking that we were going to find different genes. We knew there was not a molecular base, but we were going to be finding some recurrent genes. But the surprise was that every single patient was different. Despite that clinically they were similar, they had this aspect of patient of obese C, but only Marta had the Magel syndrome. So, Magel syndrome, sorry. So we analyzed Marta, we analyzed the father, the mother. We found this truncated mutation de novo. So we started analyzing once again, doing some tests. And thank God, because we didn't know where to go, Dr. Schaaf published the first cases just a few months later, and he gave us the answer. Because we said, okay, Marta is the first Spanish case of Schaaf-Yang. And here our work started with Schaaf-Yang in the year 2016. Some familiar faces you can see over here. We were able to receive some funds to bring you all to Barcelona, to be able to tell you a little bit about this, to grab a coffee once again and achieve this first skin biopsies and start with the functional studies. Today, I would like to seize this scientific arena to take you to our world and understand why are we so slow? Why do we take so long to do things? Because the functional studies are necessary because we need to reply to easy questions. The first one that was essential is, are we earning or winning or losing functions? This protein, which is broken, is not only not working, but also doing some damages. And this is something that we need to address. And we had to be putting these little pieces of the structure, like what's Magel for. There were many articles, many job done, as we have seen for the work of Muscatelli. And there was another work of the how that they were conducting some studies, but there was just the first hints. We knew that Magel works in the erythromer, it works together with some proteins, but these are just functions from Magel, but we didn't know much about this. So it's a basic question. When we want to do therapy, when we want to understand an illness, we need a word that scientists love, which is the biomarkers. It's basically, I need to know that this cell is sick, or this animal, or this person, whatever, and be able to see that the therapy or the intervention I'm conducting is curing, it's healing, it's improving. So for this, we need systems and we need markers, and the easier, the most trustworthy they can be, and that will be better. So finally, this is what you want, this is what you need. Well, I mean, everybody needs this, but first we need to be replying question by question. It's just not a linear thing. It's not 1 and then 2 and then 3. Everything is mixed and mingled. Studying different therapeutic targets, I can learn from different steps. The inhibition technique of Magel is helping us to understand what's happening here, but we need to take these steps. So let's go for the first things. Christian said that this is where we focused ourselves. It was basic. When you take a look at the mutations, this image is quite appealing. There were some placed here, but when we started writing to the authors, we saw there was a problem. Magel, the genome is not static when it comes to the theoretical side, and there's been some changes. So when we reviewed, the mutations were really over here. And this called our attention, like there's a barrier over here, all of the mutations are to this side. In many other illnesses, either they are accumulated in parts of the proteins that have a very determined function, they need to do something, or either they are distributed in an homogeneous way, and here we found a frontier. So we know in this tail or in this side, it's important. So this tail needs to be close to many other things to say what they need to do. When you miss this, when you lose this, they don't know what to do. So basically, we have a loss of functions. We know it, but what other things is this piece doing? The first thing we thought about is maybe they are just kidnapping these others and not letting them doing the work. But moreover, they could be doing new things. You've seen already this, Prader-Willi, we've lost the protein. In Schaaf-Yang, we have that piece, and it's something that we must not lose. Christian talked about the antibodies, and I loved knowing that there's still activity over that arena, because to be able to see a protein is necessary. To study a protein, it's basic to see it. It's fundamental to see it. Sometimes I say that it's like when you are at home and everything it's completely silent. The first thing you're going to do is check on your kids. Why is everything so silent? We need to do it, but with MAGEL, we cannot do it. We don't have the necessary tool to be able to see, which is the antibodies, what is doing the MAGEL of the cell. Where is it? What is it doing? And in which moments? And this is very important, and it's paramount, and it's the starting point. This that you see here, there's a spaghetti thing over here. It's MAGEL dose is 2. It's a model done by artificial intelligence. So these loops over here in blue, we call them alpha helix. And over here you can see this as sheets of paper, which is MAGEL 2. But this spaghetti mix here, you're seeing it's all of this domain over there, which it has prolines. It's something sticky, basically. And Dr. Weber did an amazing study. It was an amazing work. And they reached the same conclusion. It's completely disorganized. Usually, it's very difficult to have antibodies against something which is disorganized. As we are not specialists in antibodies and not even against something disorganized, our strategy is to see if we can somehow turn around the problem. I cannot do it, but I am happy that someone dares to do it. But we said, okay, let's try to somehow turn around over it or surround it. So the first thing to do is an easy model, which is the heterologist expression. It's like basically I'm going to put DNA of something into another one. I'm going to take a cell line from a tumor, which is quite standard cells. All of the labs work with the same ones. This guy gives us a standard environment, but it's very artificial. It's tumor lines. They were human, but it's not a normal cell of a patient. They are easy to work with because they grow nicely, and it's a nice tool. We're going to take this piece of DNA with this circle chromosome in which we are going to put anything we want inside. Here we are going to put this piece of MAGEL 2, and it's going to be whole, full, and then the mutation of Schaaf-Yang, truncated. This means cut. So we're going to put a piece. And to be able to see it, we're going to stick a flag or a label or a tag. And we have many of them. And against this tag, I do have antibodies. So what can I see? It's the tags. This is the first strategy. It's easy, but it's robust. So with this, the first question that we had, it's MAGEL could be wearing out. Basically, the cell detects this is like debris, let's get rid of that, but this is not always happening. So in this artificial system, MAGEL, the truncated MAGEL, is not destroyed. It's there, we detect it. Just as the wild type, the normal one. This is something concerning. I have this piece, it has an expression, it's there and can do silly things. And at least we were able to reply the first question. Christian showed this image too, is that this was a surprise for us. They are not where they should be. They're in some kind of holidays away. So in green, you see the MAGEL protein. This is a cell. Here you have 2. As they are different structures, they could be 1, 2. In blue, we have painted the DNA, the nucleus of the cell. And And the normal one, the wild type, Metal paints in green the cytoplasm, which is the body of the cell. But what's happening when we have the truncated versions? It's binded to the nucleus. It's moving to where she should not be moving. But when we do something as artificial, we have questions like, could this be due to the tag that we're using? Or the second question would be, could this be that you put the tag on the tail rather than on the head? We know that this tail is doing things. Maybe the tag is hindering some things. And you have just tried one mutation. Maybe it's just this unique to this mutation. So let's do the same, more in a better way, and trying new tags. I think it's important when different groups do different approaches because we choose different tools. And it's very important to see that we're all heading into the same direction because it's something which is not of the tag. We moved the tag from the tail to the head and we tried 6 different mutations and they all behave in the same way. Major is basically having some amazing holidays on the nucleus. And the most surprising thing is that we studied 2 variants that your kids do not have because they are the ones linked to the mortality of the babies, neonatal mortality. And this seems to be even more binded or more sticked to the nucleum. This is just an heterologism system we did in these tumor cells. We're having this tag saying, okay, this piece of protein is there, is hindering stuff. This goes in the same line of the article that Christian says. I'm not going to say the name, it's going to be a disaster, but basically they have more or less the same result. It's the same cells, the same tags, same conclusion. MAGL goes towards the nucleum. Another way of surrounding the situations or bypassing the situation is to avoid the cells of the patients that you came to donate cells or you were sending cells and to say, okay, I'm going to grab the fibroblasts of one of these patients and I'm going to inject or to place this tag. And now with this tag, I'm going to be able to see that the effect is not that much because I intoxicated the cell with the DNA, which is not theirs. But in a more natural system, the same thing is happening. This is a work in progress. We're over here. We have the edited cells, but it's important to have the isogenic controls that will be an internal control. So the patient has to turn into a control, fix the broken major, and to turn the control into a patient to have a useful tool. So along this year, we've been receiving many samples happily from you. Sometimes things had happened in the middle, but, well, you know about this. We are conducting at lines. And first, we worked with the fibroblast, is the skin cells. From this biopsy, we extract some cells which are less differentiated. They are able to grow. These cells are pretty handyman, multitasking. They can do many things. So in this work we published a few years ago, we did a test to see if MAGEL2, we know it's on the recycling side of the cell. We are clear. We know that recycling is important. Cells know about this. We know the MAGEL cells are not recycling the way they should. So, yeah, we saw there were problems in the recycling in the skin cells of the fibroblasts, and these differences were not in the Prader-Willi patients. We have many patients with Prader-Willi, and we were not able to see this. In gray is the controls, in green is the Prader-Willi, and under here it's the cells of the patients of Schaaf-Yang, which are not recycling the way they should. We also did a metabolomic study in which it's not in blood, but a metabolomic study in the cells, because we wanted to see if metabolically the cells are okay. And we see that patients of Schaaf-Yang have a reduction in the level of glutamine. We also did that transeptomic study. I will tell you about this later, but it's basically to try to listen about what they are talking about this cell. And we saw some genic unregulations, and we saw that MAGEL2 is not an important protein for the skin. Our kids do not have skin problems. So basically, it's not, the expression is minimum in the skin. It's not the ideal system to work at. We did this work because we did have the tool and it was useful to conduct all of the necessary protocols. But in the middle, our goal was basically to reach to the most effective side, which is the nervous system. So what I want to convey today is the work we did to have this neuronal model. So the first thing we did is to grab these cells from the skin and say, you're going to forget you were a skin cell, and I'm going to transform you into a pluripotent cell. You can be whatever you want to be, which is beautiful. It's important for you to remember that all of the cells in you have the same DNA. The only thing that changes is the language they speak, either if they speak in genes or in other genes. We have a gut cell, an ear cell, an eye cell, whatever. But the DNA is the same in all of the cells of your body. So we will make the cells forget that they were fibroblasts to turn them into this induced pluripotent stem cells and say you're going to be a cell or a mini brain to be able to have a system to work at, which is not the skin, but a neuronal system. We achieved this. The first steps, at least, of carrying this out. We do have a system from which we can have neurons and astrocytes. The astrocytes are the carers of the neurons. The neurons are lazy, they don't do many things, they just think. So they have to do maintenance work. And the neurons cannot grow or are mature enough if they don't have any companion. So thanks to the collaboration with Isaac, Monica, which is the doctor working in this project, has now introduced a protocol in which we have a gut culture of neurons in these cells. So the good thing is that you can control in which moment you want to analyze this. We know MAGEL is very important in this stage of the development, but the effect is going to be seen along the development too. So I can work in more early stages of differentiation or let them mature a bit more. For the moment, we've started to work over 20 days. This means that the neurons start being mature, but they are not mature enough. Same as the moment of the neurodevelopment. And here we have done omic analysis. What is this? This is going to be a genetic class. So basically, you have on the nucleum, all of you, your DNA. We all have many mutations. Nobody's safe from this, but your kids have a mutation in the MAGEL2 gene. I usually say that this is like a library, but the books cannot leave this library, so you need to take a copy. If you want to take a book away from there, you need to take a copy. What book you can rent, basically, is what makes each cell being different. So this photocopy is the RNA. This was very trendy during COVID. Well, here you have tons of it in the cell being produced. And this RNA, it's like the IKEA guidelines on how to assemble a protein, which is the one that's going to do the work. From RNA, we have the polypeptide, the protein. Here we have the MAGEL2 protein. They are going to follow the instructions. If there's an error in these guidelines, instead of having a nice chair, the chair is not going to work. These omic techniques, they study everything. It's not that I'm not going to study the transcription of RNA of one or two genes. What I want to see is everything. What's happening? Tell me in which language this gene is speaking, this cell is speaking. So neurons, over 21 days of these patients and 8 controls, we did this transcriptomic process to see what they are speaking, with what intensity and how. Moreover, with 4 patients, we did it in the iPSC and in the neurons and also proteomic. 5 minutes more. I'm clear about that. So the results of the transcriptomic is something that we received on Thursday, and they just promised that on the second one, it's going to be there on the next week. We're waiting for them. So the first thing, MAGEL2, it's okay. The expression is fine. Everything is being done. And also the imprinting. Yesterday, we talked about the imprinting. It's being maintained there correctly. This is good. It's nice because I did many silly things to them. They were fibroblasts and turned them into neurons. So the imprinting, it is what it should be. The cell lines is there. A mutation is there too. And also, adding these new pieces, we still see some problems in the RNA, in the signaling. We also see some problem of cellular binding, extracellular matrix. So we are adding new pieces to this structure to try to see what happens with MAGEL2. The other part of the project that Aina is conducting, that she's over there, it's the process of differentiating the cells to a tri-dimensional structure. Whether we make them grow with their friends of the astrocytes, they're still lonely. So if we put them in a tri-dimensional structure, we're doing some tiny brain model. It's less than 1 millimeter, but they are even quite impressive, even 3-4 millimeters, but it's something tiny. But the good thing about these structures is that they do layers between them. So we have the structure, the development of a brain. And this is more useful when it comes to study these connections. Everything has its pros and cons. You're going to see the timetable here. The experiments take 90 days. So everything takes longer, everything's slower. You kind of don't have enough patience. But the good thing is that you can choose what part of the brain you want to use, the side one, the cortical one. We could do different models. Aina has amazing little brains in which we have this good structure, well structured with mature neurons, and with these circles, which are the proliferating structures in which we have basically the parents, the ones that they are going to produce new cells or new neurons. We did this on 30, 60, and 90 days to see how the expression of a MAGEL is evolving along time, to see what moment could be optimal to work at. We analyzed them once again on this omic studies, RNA, protein, and metabolism levels, and we did a pilot test just to make all of the system work. We saw a good expression along the development. We're also evaluating longer times. We're obsessed with the imprinting. We are always going to comment on this. And okay, it's fine. It's just 1 and 1. We're not going to draw any conclusions, but we're seeing an alteration in place which are expected when it comes to neuronal development of the brain. We're seeing things that make sense. Moreover, we can see that we have all of the neuron families. It's just little brains with good structures. When it comes to proteins, we detect everything we should detect, and amino acids too. For the moment, the techniques are there. Aina already has all of these organoids, 4 and 4 for the moment, side and ventral, 30, 60, and 90 days. And when Ricardo invited me, I thought I could share the results, but unfortunately, I can just say we are working on that. We hope to have some cool things to talk about in the future. So I think collaborating between each other, we are able to answer the questions. We are going to put pieces into the structure. We bet on the MAGEL blockage or MAGEL silencing because we thought that thing should be toxic. So silencing MAGEL is the first strategy that you have to test, at least to see if we can address this. And well, thank you. I would like you to give me just 1 minute to say how thankful I am to Ricardo, Noelia, and everybody for inviting us to be here, because it's been amazing to speak to you, to be able to share with you what we do in our little castles. And I think that I hope that you understood and it was useful. Thank you.
no te...
Don't leave. Thank you very much. It was beautiful the way you explained this. Does anybody have any doubt? Any question? I have one. Fast, because we are a bit late. When you did the transfection of the 293, the cells we mentioned before, the truncated version and the wild type, do they behave in the same way? There's not phenotypic change in the cell? Yeah, we're very careful. We do HeLa cells. Okay, there's different lines of different origin, and there's many others, which are every single line. We wanted to see that there's not something in particular to that specific line. So when it comes to the phenotype, well, I mean, this is the normal thing. It's true that some proteins, when they are very toxic, when they are going to be worn out, that caught our attention. MADO is not there. It's like it's going under the radar. It's not being detected. Well, the group of Chris, it has a German name, and I don't dare to pronounce this out loud, but they saw that it's like more standing still in different groups. We need to study this, but what we want to do is to go to the fibroblast. Well, more than the fibroblast, to work with the CRISPR, with the iPSC, and see what happens. One thing is, how much are you doing the transfection? When you put this DNA, you choose. It's either 50, 100, 5,000, and this can provoke differences. So we did a huge portfolio of different concentrations, and we tried to use the lowest level not to intoxicate the cell. So maybe if we introduced more, we could see this deterioration, and, well, we don't know. And these cells, do you believe that they have some interaction with this microenvironment? Because you were talking about the matrix, the binding, if they talk to the endothelium, the astrocytes, if they modify them, it's a cumulative process. Yeah, this is the advantage of having the two models, the organoid and on the neuron. You're able to see the communications. Yeah, it's one of the experiments we want to conduct to try to analyze these communications. Well, I'm flabbergasted by everything that you've said. You've done an amazing work, and also a very difficult thing. So thank you very much. You've done an amazing work. Thank you very much. Thank you.